Innovation has been since our inception. Genetics Institute was the original innovator for recombinant technologies acquired by Wyeth. Wyeth was then acquired by Pfizer. But the commitment to innovate remains strong. The commitment to the community remains strong. The commitment to entrepreneurship remains strong. And the commitment to improve therapies, not only in this factor IX area, but also in other areas of hemophilia remains strong.
There's a small, very dedicated and focused group that continue to champion hemophilia in the company such that the needs of patients are being met.
I started first with Genetics Institute in 1986. When we think about GI at the time and look back on it now, really the roots of modern biotechnology were being formed at that time. That entrepreneurial spirit has been maintained here, within the biotechnology arm, and it's a great place to work with great people who are very passionate about what they do.
We've always made recombinant products because those are the cutting-edge technology and the safest products that are out there.
The recombinant technology was very important for the medicine in general, because the recombinant technology to allow to the patient, and of course to the physician, to have the very safe products. The other important topics about the recombinant technology is that this technology to allow to obtain the appropriate supply for the patients.
Very early on in the history of GI, factor VIII and factor IX were considered as potential products. One of the things that got in the way was the difficulty of actually cloning and making these products. This was very early in the experience base of recombinant DNA technology, and these were seen as very, very difficult things.
One of the big technical challenges in manufacturing hemophilia products is just to ensure a consistent product from each manufacturing run to the next manufacturing run.
From a compliance and regulatory standpoint, both products are made consistent with good manufacturing practices, and so that ensures consistency, it ensures quality, and we can be sure that every batch is the same as the next.
Recombinant factor manufacturing became possible when factors VIII and IX genes were discovered. By transferring the genes into host cells, scientists created cells that consistently produce high amounts of the desired clotting factor protein. Once the appropriate cells have been identified, in this case cells producing factor IX, the cell line is frozen for storage. Each of the vials could contain up to 6.5 million cells. For each manufacture run, small quantities of these cells are thawed and grown in a series of increasingly larger vessels. It takes approximately a month to grow the cells to reach the full-scale production volume. The last vessel is a 3-story tall, 6,000-liter harvest tank. Growing in a nutrient-rich but albumin-free media, the cells produce the factor IX protein. For purification, the liquid is first filtered to separate the cells and additional impurities. Several chromatography columns are then used to separate factor IX from other proteins and impurities. The columns chosen are filled with beads that effectively bind factor IX. Each column step has been optimized to enrich the factor IX while removing non-factor IX impurities. The factor IX protein is then washed from the beads using specific buffer solutions. The entire process occurs in a controlled environment and is constantly monitored. Finally, a viral filtration step is included to minimize the risk of any viral contamination. The factor IX protein is now stored at low temperatures. Upon passing extensive testing, it is processed into its final formulation and freeze-dried into a powder. The final drug vial is now ready for packaging.
Over the course of the 25 plus years of manufacturing these products, we've learned new advances in the technology, the manufacturing process, and we've also interfaced with the community itself to understand their needs. We like to try to tailor the products that really end up being the most beneficial product for the patient.
Well, goodness, I worked on Benefix since 1993. We've come up with many ideas over the years. A lot of what we work on never makes it very far, I'm afraid. I was very pleased recently when we were able to launch the reformulated Benefix as bringing forward, actually seeing in patients' hands, being able to finally bring forward some tangible enhancements. So it's a lot of people thinking and working together to come up with ideas and then actually try to make them a reality. Forward kind, right?
A hemophilia patient can expect that while we have over 12 years experience with Benefix and the clinical and safety and efficacy profile are well understood, that we will continue to invest in clinical studies to better understand the safety and efficacy of the product. Patients with hemophilia B can also expect that we will continue to improve the ease of use of products, the convenience of product. And while we've made many enhancements to our convenience of product, patients can expect that our goal is to make preventive therapy easier than ever before.
Indication. Benefix is indicated for the control and prevention of bleeding episodes in adult and pediatric patients with hemophilia B, congenital factor IX deficiency, or Christmas disease, including perioperative management. Benefix is not indicated for the treatment of other factor deficiencies, for example, factors 2, 7, 8, and 10, hemophilia A patients with inhibitors to factor 8, reversal of Coumarin-induced anticoagulation, or bleeding due to low levels of liver-dependent coagulation factors. Selected important safety information. Benefix is contraindicated in patients who have manifested life-threatening immediate hypersensitivity reactions, including anaphylaxis, to the product or its components, including hamster protein. Anaphylaxis and severe hypersensitivity reactions are possible. Should symptoms occur, treatment with the product should be discontinued and emergency treatment should be sought. Benefix has been associated with the development of thromboembolic complications, including patients receiving continuous infusion through a central venous catheter. The safety and efficacy of Benefix administration by continuous infusion have not been established. Development of activity-neutralizing antibodies has been detected in patients receiving factor IX products. If expected plasma factor IX activity levels are not attained, or if the patient presents with allergic reaction, or if bleeding is not controlled with an expected dose, an assay that measures factor IX inhibitor concentration should be performed. Patients may develop hypersensitivity to hamster CHO protein as Benefix contains trace amounts. The most common adverse reactions, less than 5%, from clinical trials were nausea, injection site reaction, injection site pain, headache, dizziness, and rash.
Uninsured? Need help paying for Pfizer medicines? Pfizer has programs that can help. Call 1-866-706-2400 or visit www.pfizerhelpfulanswers.com.
You are encouraged to report negative side effects of prescription drugs to the FDA. Visit www.fda.gov/medwatch or call 1-800-FDA-1088.